I'm going to describe to you a little bit about what bioaccumulation and biomagnification is, as well as how it applies to food chains. Because as I mentioned, we're talking about the food web of the Cooper's hawk and how it's an indicator of contamination across Metro Vancouver. And I'll give you a little bit of information about previous research that John Elliott was a part of with some of his other students, and how it sort of applies to the research that I did. And then I'll tell you a little bit about the methods and the results of, this is mostly my master's research that I'll be showing you today. So for a lot of the chemical pollutants that we looked at, these are generally known as the legacy chlorinated pollutants, and we typically call them persistent organic pollutants, or POPs. And these are generally known as the chlorinated chemicals such as polychlorinated biphenyls, or our common pesticides like DDT. And these were all generally banned in the 1970s. So for example, PCBs have industrial purposes that we use for like electrical purposes, like BC Hydro, as shown here at a substation. And then DDT was a common pesticide use that is still technically used in certain parts of the world, such as South America, as well as Africa, for malaria control. So very controlled. It's a more restricted versus a complete banned substance like PCBs. And so due to the widespread usage of these chemicals in the like after World War II, we started to see declines in birds, particularly such as the robin. And Rachel Carson, who was an investigative environmental journalist, started noticing these patterns and talking to scientists and environmentalists, and she raised the awareness about we are seeing these declines in birds due to the chemical exposure in the environment. And so then this led to the bans that we had in the 70s in the United States. And I think later on, I think it was roughly the late 70s or early 80s in Canada here. And then in the early 2000s, the United Nations came together in Stockholm, Sweden, and they came up with the Stockholm Convention, which is essentially a ratified agreement amongst these nations to either eliminate or restrict or control the usage and the prevalence of these contaminants in the environment, and to protect wildlife and human health, basically. And so this was ratified, I believe, in roughly 2016, and a lot of these chemicals that I will be showing you today are on this Stockholm Convention. So a couple other chemical pollutants that I'll be showing you tonight include the chemicals known as emerging. And this simply means that we didn't really start detecting them in samples until like the 2000s, versus the legacy ones were detected in the, say, the 40s and 50s very, very easily. So these include our brominated flame retardants. So these are chemicals that have bromine atoms attached to them rather than chlorine, like our PCBs and pesticides. And I'm also going to be talking about our fluorinated or Teflon type chemicals, like on our Teflon pans. And so these chemicals are generally used for our electronics to help them not burst into flames when they get really hot. And we also commonly use these flame retardants on our furniture, on our drapery, on our carpets in our households. And we also use them for firefighting foam agents, as depicted here. And as I mentioned with the Teflon, we use it as for nonstick cookware purposes. We also apply it to paper products for our food. So it's a very common substance. And for the perfluorinated chemicals, we also use it for stain fighting or waterproofing our clothing, such as Gore-Tex fabric. That's all with perfluorinated chemicals. So a unique thing to all of these chemicals that I just mentioned is that they're all very persistent, so they don't break down easily in the environment. So the sun and water and other physical aspects don't break them down easily, and they tend to bioaccumulate, meaning that they get absorbed into the tissues of the organism, such as the lipids. So generally, all of these chemicals get stored in our fat tissues. And they're all inherently very toxic as well, these chemicals. So at very low dosage, they can cause, say, lethal effects causing death or potentially sublethal effects, so causing like disruptions in our hormone levels. So the way we get exposed to these chemicals is either orally, like say through drinking as depicted here, or through our diet, or we're breathing in these chemicals, or we can be dermally exposed to these chemicals. And so through these different exposure pathways, the chemical builds up into our tissues, and we do have some capability of eliminating these chemicals, like metabolizing them out of our system, but the portions that don't get metabolized out, we store it into our lipid tissues. So as seen here with this adipose tissue, this is giving you an example of where these chemicals are often being stored in our bodies here. And so, sorry?
Oh. That wasn't us, that was some interference from outside. We don't know what that is.
Oh, okay, I'll keep going. Okay, and so when you have the chemical being stored in the fat tissues, and then you have another animal that comes in and eats that subsequent animal, what then occurs is a process called biomagnification. So then we start to see that the chemical at these lower levels of the food chain, as we subsequently go up each trophic level, the concentration of that chemical will increase going up. And so this is a type of bioaccumulation called biomagnification. And so this is also a very common occurrence with these chemicals that I've shown you. So in Canada and the United States, we both have very similar regulatory criteria or metrics or laws that we use to control these substances or to evaluate their potential to cause harm to wildlife or ourselves. And so we even use very similar parameters in Europe as well. But all of these metrics that we tend to use are all based on data collected from aquatic organisms generally, such as the salmon as depicted here. And because all of this data has been generally based on fish a lot of the time, it's not going to be very appropriate usually for our air-breathing organisms because it was all based on fish, which typically breathe water. And so a lot of the properties of these chemicals will actually behave differently in an organism that breathes air instead. So that's where a lot of my research comes into play here. So we wanted to focus on an air-breathing organism, the Cooper's hawk. And so some important aspects of the Cooper's hawk for you is that it has a wide distribution across North America. I'm sure some of you are very familiar with this species. It primarily eats birds, even though I know a lot of you know that it eats rats and other critters and such in your backyards. It's also known as a year-round resident here in Metro Vancouver. So this is also a big reason of why we chose to focus on it. So then it's representing local exposure versus being a migratory type species. And it also is known to have very high nest site fidelity. And so this is really important when I show you the previous research done by other students. And it's also a bird, an urban bird that's been very well studied, such as by Rosenfield, as I'm showing here in some of these books that are here. So as most of you are probably familiar, it's part of the accipiter family, which includes our sharp-shinned hawk, the Cooper's hawk, or our northern goshawk. And so a previous student, Jay Brogan. Oh.
Being spammed by somebody.
It looks like,
yeah, it looks like somebody's breaking into our site here. Let's see what's happening. I don't know.
I can just keep going. I think it was the iPhone that logged in, and they've muted themselves now. So
okay.
I sound a little better. So Jay Brogan was a previous student of John's, and these were all of the nest sites that he investigated across Metro Vancouver, all these little black dots. And he collected, he did blood work on Cooper's hawks in 2012 and 2013. And so he sampled 22 nest sites, and these are very similar nest sites that I then went to for my research. And so for his work, he collected 21 adults and 15 nestlings and collected a blood sample from each of them. and then looked at thyroid hormones and chemical concentrations in the blood here. And so just to show you a little bit of his data here on the left here with where I have adults, this is showing the PCB concentrations in the blood of adult Cooper's hawks versus the TT4 or thyroid hormone in the blood. And we can see that as the concentration of PCBs increased in the blood of adults, that basically your thyroid hormone levels, your very important T4 levels, were going down steadily. And then we see on the right here with the nestlings in the blood, basically as the concentration of dieldrin, which is another type of pesticide similar to DDT, we basically saw that as the concentration levels in the nestlings increased over time, that there was fewer nestlings being fledged per active nest site. So this is just showing you an example of what these chemicals have done in this indicator species of the Cooper's hawk. And some other research done by John and Jay Brogan here, they basically collected dead carcasses of Cooper's hawks, and they analyzed the liver tissue for these flame retardants and other legacy contaminants. And they found that one particular bird in Langley near the landfill was considered one of the dirtiest birds in the world, or found in Vancouver here. It had it had the highest levels of flame retardants reported in the literature to date. So some people were saying it was so toxic, it was flame proof. So this led my supervisor, John, to ask the question of how are these birds getting so contaminated? And so this led to wondering about the food web and its diet that it's eating. So we wanted to use the hawk as this bioaccumulation model sort of indicator species. And we wanted to assess its various exposure routes, such as through the diet or through dust exposure on the feathers where it would preen, and would that be another potential exposure route? So the main study goals for my research was measuring the concentrations of these various contaminants that I've shown you in a food web that included the Cooper's hawk. And then we would quantify the amount of biomagnification occurring in the food web. And then we also wanted to compare what goes on in a terrestrial food web to what has been previously done in aquatic food webs and see what kind of differences there were. So this is showing you an example of the food web of the different species that I collected samples for. We have the Cooper's hawk at the top. We have various passerine species, and say a woodpecker here with the northern flicker that it's known to potentially eat and predate. And we also included several different types of invertebrates as well. And we even had a type of berry or vegetation that these birds might also be eating. And so for my food web, very similar to Jay's work with his nest sites, I actually used a lot of his previous nest sites. I would go out to them and check them for activity. So all these little dots here are the active Cooper's hawk nests that I sampled, and the purple circle around it represents like the assumed nesting territory of the bird around the nest, of where they would potentially maybe be collecting prey. And so that's where I tended to try and focus my sampling of the other organisms of the food web. So for finding nest sites, I would go to these previously historical nest sites that Jay gave me, or other people such as yourselves in naturalist groups. I would contact them and ask them, hey, do you guys know where any active nests are? I'm doing this research project. And I would go out at early morning before dawn and play Cooper's hawks. With my little trusty Fox Pro here. And I would do the dawn chorus sort of sequence where you play it for, I think it's roughly 30 seconds, and then you wait a minute and you wait for a response basically. And so this was quite effective to determine if these nests were active, and it was also an effective way for me to find new nests that hadn't been documented yet. And so up here, you can see one of the active nests that we collected. I believe this is somewhere in Vancouver. It might be Yukon Street. I can't quite remember here, and Then we would climb into the nest. I would either hire a climber or we would use a bucket truck to collect one egg from the nest. And I also wanted to participate in this, so I also climbed one tree to collect an egg myself as well. And to give you, here's an example of what it looked like in one of the nests. So according to our permit, we were only allowed to collect one egg from a nest as long as there was at least 4 eggs in the nest. So that, say there would be like, say 3 eggs left over if I collected an egg. And basically in all of the nests, we had at least 3, if not 7 eggs in a nest. So they were doing very well in 2016 when I did this egg collection. And we also went back to all these nests throughout the year in 2016 to monitor, was there any abandonment or was there any issues from, say, collecting this egg? And basically there was 2 to 3 chicks fledged per nest, indicating a 94% reproductive success. And this was actually very similar to Jay's work. He had roughly a 90% reproductive success. So this was considered very high and a fairly good amount for our area here. And so this is an example. I'd crack that egg open and put it into a jar, and then we'd send it off for analysis, basically. And so, as I mentioned, we also collected a lot of passerines or songbirds. So according to work done in Victoria, where they did nest monitoring, they found that Cooper's hawks in the Victoria area were focusing their predation on starlings, house sparrows, and American robins. And so we were going to focus our attentions on these 3 species as well. So potentially trying to do mist netting or collect eggs or nestlings from nests. However, in the Metro Vancouver area, that's often very difficult to do because there's so many people around it. And so it's proving very challenging. This is an example of a robin nest where I collected, say, a nestling from, and say an egg example here. Basically, because this is proving very challenging to collect enough samples, I reached out to Wildlife Rescue Association, and I basically went to their mortuary freezers and then was able to collect numerous other species that the Cooper's hawk could potentially be eating, such as the dark-eyed junco, towhees, various thrushes like the very thrush or the hermit thrush. So basically I was able to expand my species list easily because I would collect these dead species that had either died on arrival or died of their injuries. And then I would coordinate where they were picked up from with my nest sites, basically. So I would only select the ones that were close to my various nest sites and use them. And so, as I mentioned, I wanted to collect species or food items that these birds may be eating. So we put little pitfall traps out near all my nest sites. And so right here, you see it's underneath some shrubbery or these ferns in the hopes that it'll not be disturbed by people or the public. And so this was quite effective. And we were able to collect lots of different beetle species, such as the bronze beetle or Carabus nemoralis, as well as the sow bugs or pill bugs as depicted here, or like millipedes and centipedes. So we had a lot of different invertebrates just collected in those little pitfalls. And we also collected earthworms. And to do this, we would set out a grid and we would make up this solution of allyl isothiocyanate, or AITC, which is essentially just a chemical derivative of like mustard plants. And so very similar to the pungent flavor of wasabi or mustard, that's what AITC is. And we would mix this with isopropanol and water and spread it on the ground. And then the earthworms would all emerge out of the ground, kind of like Night of the Living Dead zombies. And then we'd pick them up and wash them off in some water. And same thing, we would then put them in jars and send them off for analysis. And I mentioned we also collected a berry or a plant representative that these birds might be eating. So we focused on Himalayan blackberry because it's so prevalent across Metro Vancouver, very easy to find at all the different nest sites. And we would collect these ripe berries for analysis. So once you have collected all these samples, we actually have to process them in order to be able to analyze the chemical contamination in the tissues. So to do this, as I'm showing here, some of this hopefully it doesn't bother too many of you, but you'll see some dead animals here. We would have all these little birds and we would have to pluck all their feathers off. And then we'd have to chop them up and they're all kind of frozen or semi-thawed. And then chop them up into like a kind of a mix and randomly select the mix. And we'd have to pulverize it all up to make it into like a homogenized paste, which we would then analyze at a lab in Ottawa. And so for these chemicals, as I mentioned, we would mush up all those samples and we would then run it through these machines called a gas chromatography, mass spectrometry. And this was for the chlorinated type chemicals. And then for those Teflon type chemicals, they use another type of machine called a liquid chromatography. So these are very expensive machines. I didn't actually have to do this analysis, thank God, because that would be really complicated. But we had some labs do this for us. So now to show you some of the results of all this work that we did. So for one of the main contaminants, the PCB 153, this is one of the PCBs that had the highest concentrations in all of my samples. And it's known to consistently biomagnify in all the different types of food webs that it's studied in. So it's a really good chemical to look at because then it's kind of like a positive control. Like if your study isn't able to show that this chemical is biomagnifying, you've potentially done something wrong. So this one is a good one to look at. And some things that, as I mentioned before with these PCBs, they're known to have significant effects to birds or to other organisms for endocrine hormone disruption. So as I'm mentioning here, it reduced hatching success, delayed egg laying. It can also decrease being consistent for incubation as well. So it can have a lot of potential sublethal effects versus say a lethal effect. And so here's a plot. We have the species going up in trophic level along our x-axis to our Cooper's hawk here. And then on the y-axis is the average concentration of the chemicals in each of our species. So to help you sort of visually see which species are in sequence here, we see that like close to 10,000 nanograms per gram of chemical in the lipids. This is, I'll point out that this is grams per lipid. So we've normalized these concentrations to the amount of chemical in the fat tissues of these organisms. So we see that Cooper's hawk is very high, so indicating this very strong relationship or biomagnification occurring in the food web here. So this was good. Our study did a good job because we saw that there was biomagnification. And now looking at one of our pesticides. So DDE is actually the metabolite of DDT. So when you absorb DDT, it can get broken down into DDE. So metabolite, this is actually the chemical that has that very strong relationship with eggshell thinning versus it's not actually DDT. And this is the contaminant out of all my contaminants that had the highest concentrations out of, so it was even higher than PCB 153. So as I will point out here, the same sort of graph as I showed you earlier, here's the highest level is up to 100,000 nanograms. So we can see with our Cooper's hawk, it was higher than that PCB 153. So again, though, we see like this strong trend of the concentrations going up as we go up the food chain here. And so for one of our flame retardants, this is BDE 99. So basically some of these types of flame retardants can get broken down into smaller sized contaminants. So this is an example of one of them that is commonly found in the environment. So this type of flame retardant is one of the ones that's most commonly detected. And I think it also had some of the highest levels of out of the flame retardants in my samples. Oh. Sorry about that. Where'd my slide go? Ah, geez. are you guys looking? Oh, I went all the way to, sorry about that. Let me go back to the slide we were at here. There, okay. You guys can see that. Apologies, I don't know what happened there. So out of the different flame retardants, this is one that's generally considered more toxic than some of those other flame retardants. And so similar graph here, I'll point out that here it's indicating 1,000 versus with PCB 153 and DDE, that was again much higher. So it's generally a lot lower than those other two that I showed you, but still very high. And so again, we still see that very strong relationship going up the food web, these concentrations going up. And then showing you some information for PFAS here, which is one of those perfluorinated or Teflon type chemicals. It's one of the more well-studied of those Teflon type chemicals. And same thing, it had some of the highest concentrations compared to those other perfluorinated chemicals in my food web. And it's also associated with reduced hatching success and as well as embryo mortality in birds. So pointing out some of the concentrations, this is actually on wet weight. So we didn't actually look at the chemical concentration in lipids this time. So this is just overall in the organism. So again, we still see this very strong trend that at lower levels of the food web, it was a lot lower than say up to the Cooper's hawk. So still indicating very strong biomagnification occurring here in the food web. And so for some comparisons, with aquatic food webs, we saw that for our PCBs and our pesticides, biomagnification levels were actually quite similar versus with flame retardants and the perfluorinated chemicals, we actually saw biomagnification in our terrestrial food web tended to be higher than in aquatic food webs, particularly ones that didn't also have air-breathing organisms like the seal as depicted here. And when we looked at other terrestrial food webs, so there's generally been only one other terrestrial food web that's been done, and that's the lichen caribou wolf food chain system. So in comparison to that type of food web up in the Arctic, pesticides, particularly DDE, we had much higher biomagnification in our avian food web down here in the south than compared to the mammalian one up in the Arctic. And we also saw that for flame retardants or the PBDEs, we saw same sort of thing, biomagnification in our urban food web with birds was generally a lot higher than in this mammalian food web in the Arctic. And potentially this could be because we're looking at a remote northern system versus our more urban system down in the south, which we would probably generally have higher levels of pollution down here. Could also be that mammals sometimes have more types of enzymes in them that allows them to eliminate some of these chemicals easier than say for birds. And also, I forgot to mention for those perfluorinated chemicals, we also saw higher biomagnification in our bird species than say in those mammal species up in the north as well. So very similar sort of thing as I was mentioning. This could be because of enzyme differences. Also, birds tend to be able to digest their food a lot more thoroughly than a lot of mammals do. So oftentimes, I'm sure some of you are all very familiar with whitewash. Birds are very good at digesting all of their food really, really well. And so because they can do that, they're actually able to absorb more chemical that way into their system. So to reiterate what some of the main findings were of our research, we saw that these legacy and emerging persistent organic pollutants are still very much present in the environment down here in Metro Vancouver. And we actually saw that biomagnification in our avian food web or terrestrial avian food web was either very similar or actually higher than in compared to some of these aquatic food webs or in that Arctic terrestrial system. And so strongly indicating to us that birds are likely a very more sensitive taxa than compared to mammals, and are hence very good indicators of contamination and the health of your environment when we look at them. And so basically, this is also just emphasizing that we really need separate regulatory criteria for our air-breathing organisms such as birds compared to fish. They're not apples to apples, it's more like apples to oranges. So we should really have separate criteria evaluating them separately. So now what? Now what do we do with all this information that we know? And what am I currently doing with my PhD work? So a lot of this work is being implemented into my PhD research. So basically the people that use this data that we collected is the government, such as the federal government. Even industry can use this information, particularly for risk assessments of sites that maybe are planning to do mining or housing development, etc. Other academia people can also use this information to build upon the research. And some of my plans are to construct regulatory guidance and criteria on how best to assess biomagnification of chemicals in terrestrial organisms and food webs. So that's basically what I'm planning to do, is help create legislation for this. And however, some of you may be wondering, what can you do personally as an individual? So some things that we can all do is use less, and especially try and use less plastic if we can. And if you do have things you need to get rid of, it's dispose of it responsibly. So you can actually go to recycling.Metro Vancouver, and they'll actually tell you various options of how to responsibly get rid of a lot of your items you want to recycle or throw out. And buy organic food when possible, because we'll generally be using less pesticides. And so the environment is then getting exposed to less pesticides through organic farming techniques. And you can also support changes to the Canadian Environmental Protection Act. You can actually go here through this website, or if you go to CEPA, like the .gc.ec, I believe, something like that, you can look up information about CEPA and or email your member of parliament and stress that you're really wanting changes to the Environmental Protection Act, or like stronger legislation regarding chemicals and the management of them. And I would just like to acknowledge all the various people who helped make this project happen, and the work. I couldn't have done any of it myself. So I had numerous people helping with, say, field assistance, as well as laboratory assistance, and several places that did the actual laboratory analysis. So thanking all those people and my collaborators. And thank you guys for your attention and your patience with the little glitches that go on here. And am happy to take any questions if you have them.
Thank you very much, Kate. I'm not muted. Thank you very much. That was most interesting. A little bit disturbing, but most interesting.
Yeah.
So we'll open it up to the floor now. Has anybody got questions for Kate? Speak up, because I think she should be able to hear you, because, well, I guess this is the speaker, but if you don't hear the question, I'll repeat it.
Oh, excellent.
Dennis.
I was just wondering if there were any nests out all the way out in Chilliwack at all, or something else is
Dennis wonders if you did any studies in any of the Cooper Cox nests out here in the Chilliwack area.
Have they been studied? Like in the nests? No.
Was it part of your study?
Was this area part of your study, I guess?
No, unfortunately, Chilliwack was not part of my study area, but I would suspect that you would have very similar levels as to what we saw in Metro Vancouver. You might potentially have higher levels of some of the pesticides like DDE, because, for example, Richmond area, if I can show you what some of the concentrations look like for soil. So when we collected soil samples across Metro Vancouver, we saw that, say, Richmond and Vancouver, like the southern part of Vancouver, tended to have higher levels of DDE in the soil than some other parts of Vancouver. So I would kind of suspect that areas like Chilliwack or the Fraser Valley might also have higher levels of DDE in the soil, and thus probably up the food web because of using it in the past out in the Fraser Valley. But I would probably suspect that the contaminant levels of like flame retardants, like the BDE 99 here, or polychlorinated biphenyls like PCB, would probably be fairly similar to what we saw in the rest of Metro Vancouver. But I would suspect, yeah, pesticides might be a bit higher out in the Fraser Valley. And hence probably a little bit higher than samples that I collected in Metro Van. I potentially think in John's work and Jay Brogan's work in some previous publications, some Cooper's hawks in the Abbotsford area tended to have very high levels. And like I was mentioning earlier with that bird collected out in Langley near the landfill, it had some of the highest levels. So again, that wasn't included in my research study, but there's been some reports of very high levels out in the Fraser Valley though. So could potentially be high in Chilliwack as well, maybe. I hope that answered your question.
Yeah, exactly with that. Any other questions? The people on Zoom, I don't know what's happening with the people on Zoom. Let's see what's happening. I wondered, Kate, and myself, about the eagles that all feed at the landfill as you go out to the ferry on the highway. I don't know what number highway that is, or something, or whatever. I'm just wondering, has anybody ever studied the bioaccumulation in those birds?
Yes, my supervisor, John, has done many years of research on the bald eagles in the Lower Mainland here. They also have nest boxes of starlings out at the landfill. And I believe that some of that research shown for the starlings out at the landfill is that generally birds near the landfill compared to more rural areas like say the Fraser Valley, the birds near the landfill had higher levels of contaminants, particularly like flame retardants, than compared to the farming areas. So I would suspect that birds like the eagles, or say the gulls that tend to frequent the landfills, could have quite high levels of these contaminants, such as the flame retardants in them and such.
One of our members is asking, where are those flame retardants coming from? Like, why are they out there in the environment?
So what happens when we make our various products, like our furniture or our electronics, our carpets or upholstery, basically these chemicals are sprayed onto those fabrics, and they're not actually really strongly bonded to it. And over time, they can slough off the product, particularly like plastics and things like that. And so because they're not strongly bonded to it, they come off into the environment, but then they don't break down easily once they're removed or sloughed off that product. So when we drop off our various items to the landfill, these chemicals basically slough off the product and then build up in the soil. So they bind to like the organic matter or the organic carbon in the soil, and then up through the food web. So through those earthworms and such, they absorb it because they're eating all that organic matter, and then subsequently up the food chain it goes, basically. So it's because we love to put these chemicals on everything, because we don't want our houses to burn and we don't want our computers to burst into flames and things. So it's just so ubiquitously used. That's often why we see it in the environment so much, even though now we're since banned it. I will point out though that since we've banned these chemicals like the PCBs and these flame retardants, and my supervisor, John, who continues to study various organisms. Like, so for example, with seabirds, they've been studying them since like the 70s or 60s, these types of contaminants. Basically, over time, we started to see declines in these chemicals in the eggs. So by banning them, the chemical does slowly decline in over time. So, but they're still technically out there in the environment. We'll probably still always have them because they're so persistent, but they slowly do kind of dissipate over time. So, because that's what we're generally seeing in those egg samples from seabirds over time. So we did see that there was an effect of banning these chemicals, basically. So that's a good news story.
Any other questions?
See, I'm sorry. So these chemicals are banned, but because they are so persistent, they still here even when we are not using them.
That's right. Yeah.
Wow.
But as I did mention with DDT, other reasons why we still see it at quite high levels is potentially because we're still allowed to use it in certain countries for malaria control because it's so effective. And because we have these chemicals undergo something called long range transport, meaning that wind and ocean currents can move that chemical, like say where it's used in Africa or in South America, and move it up to say the north or to the polar regions basically. And so that's why we often see such high levels in the Arctic where we don't even use these chemicals usually. So
yeah, that's why it now has sense how they are everywhere. Yeah,
yeah,
thank you.
Yeah,
but
it's kind of depressing.
Yeah.
Thank you, Mercedes. Thank you, Kate. Another question, or Heather, what's your question?
You mentioned that it's like the federal government about your concerns. If you're going to write somebody in the provincial government, who would be the best person to write this community or which department?
Could you hear that question, Kate? No, she's asking, and so you gave us an address or a link there to write to the federal government of who in the provincial government would be the person to complain to.
Oh, good question. Maybe the Minister of Environment, maybe. I'll look into that and I'll send you an email of who you could maybe talk to. But SEPA is generally federal, but there may be people at a provincial level you can call and complain to, because for example, chemicals such as rodenticides, maybe some of you are aware of, can cause sort of similar effects where we can see them bioaccumulate in organisms. Like when an owl eats a dead rat that's eaten a rat poison, we can see it accumulate in its system if it keeps eating these poisoned rats. And so that has led to like a ban of some of these rodenticides. And that was essentially done by people such as yourselves and myself, calling upon our municipal governments and our provincial government to not use the chemical. So calling and complaining, sending emails really does work. So, because that's an example right there of with the rodenticides. But there's probably specific people at the ministry that you can contact. So I can look into that and then send your group an email of like follow up.
That would be nice. And could you send us the links that you had up on the screen tonight? Because I didn't have any way of capturing them on a computer. It's not my own acceptor. So if you could send us all those links in an email, then
yes,
we'll share them with the membership.
Yes, excellent. Yeah, I'll totally do that. Yeah.
Thank you very much. I had one other question. If there's anybody else out here with a question, Bianca? No. I was wondering about, everybody's interested about raptors eating rats. Have you done any studies on how serious that problem is? How many raptors taken into rescue centers are actually victims of the bioaccumulation of rat poison?
Oh yeah, lots.
Yes.
Yes. So again, my supervisor John has done lots of work looking at that over the last 20 years or more, and I actually did a risk assessment for my local municipality of North Vancouver, looking at the potential risk of using these rodenticides at rec centers and how that could potentially impact owls. And so using data from John of owls that are collected across Metro Vancouver and analyze, like their livers are analyzed when they've died for these rodenticides. I basically, through my risk assessment, found that there's potentially enough for like rodenticide usage across across North Vancouver in particular, that it could pose a risk to owls. So I, through that little risk assessment, I let Elise Roberts know, who is a North Vancouver resident, very concerned about this trend, basically, of owls, dead owls being found near rec centers. And so through that risk assessment and her concern, she went to the District of North Vancouver and encouraged getting a ban in North Vancouver implemented here. So it very much is a risk, like raptors eating, say, poisoned rats, because particularly raptors that are near urban areas tend to show a high trend of eating rats, basically, because rats are so higher in density in urban areas, basically. So there's this strong correlation between like owls eating rats and rats being higher in density in urban areas. So then increasing the potential that they're going to become poisoned by poisoned rats.
Well, thank you very much. Any other questions? Or that, Kate, so I'll just say thank you very much, Kate. If you could maybe on an email send us your mailing address so that we will have that, rather than me trying to write it down correctly now. If you email, I'll get it right. And,
Totally, yeah.
we're very much, I very much appreciated you coming out tonight, putting up with all the technical difficulties, but that happened. And so if you want to sign out, that's all right. But the people that are on by Zoom, please stay, because we've got some announcements and some announcements and things to sort out here for the Nature Chilliwack. So thanks again, Kate.
Excellent. And well,
Thanks,
if
Kate.
anybody has questions that they think of later on, feel free to email me your questions. I believe Jan and Lee have my email. So if you guys want to share it with people, if they have questions, or if they want to find out who to contact, or they want to know the chemical that I use to collect worms for fishing, please feel free to email me. Yes,
Okay.
and thank you guys so much for inviting me. I really enjoyed talking to you tonight.
Thank you.
Yeah.
It's great.
Bye-bye.
Have a good night.
You
Bye.
too.
Bye.